CRISPR-Cas9 is a new technique for gene editing. In this episode, we'll learn about the applications of gene editing, methods of gene editing. We'll talk about CRISPR-Cas9 in details and how it is used in gene editing. This is Alia Aracha and you're watching Denable. Stay tuned. Why gene editing? Why did scientists think about gene editing? It's applied in many fields, such as scientific research. For example, to delete or stop the action of a specific gene to know its function. Second, it's used to increase the quantity and the quality of the crops. For example, GMO corn. GMO stands for genetically modified organism. Third, in treatment of some diseases, this is known as gene therapy. So gene therapy is applied only on limited number of diseases. For example, spinal muscular atrophy, which is a disease that is treated by gene therapy. Methods of gene editing. The oldest method in gene editing is to use restriction enzymes. Then we had zinc finger nucleases, TALEN gene editing, and finally CRISPR-Cas9. Now scientists are trying to develop new methods based on CRISPR-Cas9. CRISPR stands for clustered regularly interspaced short palindromic repeats, and Cas9 is a nuclease which cleaves the DNA. This figure shows the structure of CRISPR-Cas9. This genome is a bacterial genome, and these are the spacers found between the short palindromic repeats. Here, the Cas9 gene, which encodes the Cas9 protein. Cas9 protein is a nuclease that is necessary for DNA cleavage. In 2020, Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize for discovering the biochemical pathway of CRISPR-Cas9. So that was a very important event in scientific research. Note that we say discover because gene editing by CRISPR is a natural process that occurs by bacteria. Doudna and Charpentier described the pathway of CRISPR-Cas9 as follows. Naturally, bacterial cell uses this mechanism of action against the infection of bacteriophage. So when a bacteriophage comes to infect a specific bacterial cell, it ejects its DNA into that cell. As a defense, the bacterial cell produces nucleases to cleave the viral DNA. After cleavage, the bacterial cell takes a segment of the degraded viral DNA into its genome. So a piece of the viral DNA is now inserted into the bacterial genome as a spacer in the region of the CRISPR. This is called the CRISPR region. Bacterial genome naturally possesses the Cas9 gene and type 2 CRISPR locus, which encodes the tracker RNA. When the bacteriophage comes to infect the same bacterial cell again, this complex starts to be transcribed. So type 2 CRISPR locus is transcribed into tracker RNA. Cas9 gene is transcribed into the Cas9 protein, which is a nuclease, and the CRISPR region is transcribed with a segment of the viral DNA. When this segment is transcribed, we have a complementary strand to this DNA segment and is also complementary to the viral DNA segment. So the CRISPR RNA sequence is complementary to the viral sequence. So it's necessary to guide the Cas9 into the viral genome to be able to cleave it. Tracker RNA is necessary as it binds to the Cas9 enzyme and it triggers the CRISPR RNA to it. Now we have a complex of the CRISPR RNA which guides the complex to the viral DNA and the Cas9 which cleaves the viral DNA. On the viral DNA we have a PAM site. This PAM site is necessary for Cas9 to recognize the viral genome and start to cleave it. So CRISPR-Cas9 complex consists of CRISPR RNA, Cas9, tracker RNA. CRISPR-Cas9 directs the complex into the viral genome. And in case of gene editing, we say it's complementary to the target gene, so it directs the complex to the target gene. Cas9 recognizes the PAM site of the viral genome or target genome in case of gene editing, and it's the enzyme responsible for DNA cleavage. Tracker RNA binds to the Cas9 enzyme and the CRISPR RNA, so it guides the CRISPR RNA to the Cas9 enzyme. CRISPR RNA and tracker RNA are called small guide RNA. CRISPR-Cas9 in gene editing. Same complex. This is the target sequence of the genome. CRISPR RNA, tracker RNA, PAM site, and the Cas9 nuclease. By binding this complex, cleavage can occur at a specific site. The cleavage is called site-specific double-strand DNA break. After double-strand DNA break, we have one of three choices. First, disruption. The gene sequence can be disrupted by a process called non-homologous end joining. This process results in addition or deletion of any base pair, disrupting the original DNA sequence, and this causes gene inactivation. Second, deletion. If we have CRISPR-Cas9 complex here and also at this site, we have cleavage at both sites. So this segment can be deleted and then rejoined by non-homologous end joining. Third, correction or insertion. So by adding a DNA template with the CRISPR-Cas9 complex, this allows the cell to correct that segment with the added segment, or even can be inserted instead of it, and joined by a process called homologous recombination. Techniques developed from CRISPR-Cas9 complex. Scientists are trying to develop new techniques for gene editing based on CRISPR-Cas9. For example, some scientists could create a segment of DNA into target genome without inserting a template and without making a double-strand break. In this case, CRISPR-Cas9 complex consists of the prime editing guide RNA, which is the guide RNA, and a specific RNA sequence. Also, we have impaired Cas9 enzyme that cannot cleave DNA, and a reverse transcriptase enzyme which reverses the RNA into DNA. So by adding this complex, the reverse transcriptase enzyme can convert this RNA sequence into the required DNA sequence at a specific site with the help of the guide RNA without causing any cleavage because the Cas9 enzyme is impaired. These are the references, and thanks for watching.